Yeah.
This is not a priority.
No.
The defense.
Okay. Yep. No, it's already broadcasting.
Ok.
Yep.
You ready?
Yep.
Ok. Bonjour. Bonjour tout le monde. Donc, vous êtes bienvenus à assister à la soutenance de Paula Sanz-Leon. On va commencer. Je passe la parole directement au président du comité d'Europe.
Bonjour, et merci d'être venu. Donc, je vais introduire le séance de thèse de Paula Sanz-Leon. Donc, je vais commencer par présenter le jury. Donc, Gustavo Deco, de l'Université Paula. Klaus Hildetag, von Hamburg. Andrew Davison, de clinique à Gilles-sur-Yvette, c'est Paris, et Victor, qui est le directeur de thèse. Donc, Gustavo et Klaus sont les rapporteurs de cette thèse. Donc, Paula, tu as 45 minutes pour présenter ton de thèse, et après, nous passerons au questions du jour.
Merci, Laurent. Well, welcome everyone, and thanks for coming. In these 45 minutes, I will present, which I did during this at the Institut de Neurosciences des Systèmes, under the direction of Victor Gersa. And my thesis is entitled Development of a Computational and Neuroinformatics Framework for Large-Scale Brain Modeling. First, I would like to thank again to the jury, Andrew Davison, Gustavo Deco, Klaus Hilgetag, Laurent Parinet, and Victor Gersa, for your time and interest, and for coming all the way to Marseille. In this presentation, I will describe the computational model that has been implemented in the neuroinformatics platform, and then I will briefly discuss an ongoing project about the space-time structure of the couplings, or the space-time structures of the connectivity matrix. And then we will go to the conclusions and then future and challenges for my work. The computational model, discrete brain network models. We are in neuroscience, and in theoretical neuroscience we have certain models, but for every science, like neuroscience, we have science as well, which is a particular realization or an implementation of a model. What I will describe here is a particular notation of how we build this brain network model. With really feature of which is topic models for assemblies is a state variable that we call n. But only feature, so we need more state variables. If we have more state variables describing a certain population, we go to a single neural mass. The mass framework was initially used in the 70s by Walter Freeman, And it was introduced to have a reduced representation of population of neurons. Then we have this particular notation that includes many states variables of a certain population. But as we know, in the brain, we have different types of cells, therefore different types of population of neurons. Then we need to keep adding state variables, and we need to keep adding neural masses. If we connect two neural masses now, we consider this in our computational framework the minimal neurocomputational unit, yes, that is going to describe the dynamics of the brain. Now the question seems a bit more complicated, but it's only to describe sometimes the nonlinear coupling that may exist between these two neural masses. And in general, we can give a functional character to every neural mass. For instance, this could be an excitatory neuron, and this could be an inhibitory population of neurons. Now, let me introduce you to the notion. We call this level V0. This level V0 is describing the interaction between two neural masses. But then, as I was saying, we are interested in mesoscopic models and in large-scale brain models. Now, we introduced the idea, the concept of level V1 and V2. And what are describing this? Two different types of connectivity. Structural connectivity. And the level V2 represents the connectivity or neurons. Two different populations, a framework. Why is particular notation important, particular building up important? Because we want to implement many different models that we find in the literature, and we can actually exchange models, and so try to standardize a notation. We can frame different models into a common description. And we have all the different connectivity levels, V0, V1, and V2, into one single equation. This part corresponds to the neural mass model, which gives the particular dynamics of a node in the network. Then we have noise or external inputs, stimulation, and we have this big term here, which represents the coupling term. This coupling term means all the activity that comes from the rest of the network into one particular node. And this variable set could be a linear or non-linear transformation, depending on the model we are dealing with. As people working in this domain may know, there are mainly two different types of models, voltage-based and activity-based, and they say they are not quite equivalent because we are describing our implementation. We show that we can include both type of models in one particular description. What we also did here with this particular notation was to define the complexity of the system. Sometimes we speak about complexity, and the system is nothing else than the number of equations that we have in our network. And is the number of nodes in the network, and n is the number of state variables of a particular neural mass model. M is the number of neural mass models we have. O is another dimension that we have in some particular models, which are called multimodal models, and capture the features of a population. To summarize, this table here, which I included in the manuscript, it's a summary of the different models that have been implemented and the different models that have been widely used now by the community for network models. It includes the Kuramoto model, the one-on-one model, which it has an equivalent in the dynamic mean field model, a generic 2D oscillator where we can actually capture, we can represent the Fitzhugh-Nagumo dynamics with Van Pol oscillator as well, Wilson and Cohen, Larter Breakspear, we call it, which is a derivation of the Maurice-Lecar neuron model, a field model, Janssen and Late model, and the Stefanescu-Yersa models, which are the multimodal models I was speaking before. As you can see, there is a high variability in the dimensions of these models. In the masses they have, sometimes they represent, as I was saying, excitatory, inhibitory population nodes, and in the number of state variables that each neural mass has. Furthermore, there is another complication that having such a variety of models presents. It's the coupling term. The interaction between a particular node and what it sees or what it gets from the rest of the network, and sometimes it could be a nonlinear transformation, directly a linear transformation, or in this particular case of the Kuramoto model, is the sign of the phase difference between oscillators. So all these different models, in fact, can be fixed into the general equation we presented before. Levels we were speaking. This actually corresponds to structural connectivity. The level V1 is in fact the intracortical connectivity. So those fibers or lateral connections, those fibers going inside the cortex. While the level V2 represents the connections between distant regions, yes, and they correspond to the Y fibers. Use system, it could be action fibers, or it could be between really the occipital lobe and the frontal lobe, and the connections between the cortex and the subcortical structures. Now, this nice figure made by Andreas Spiegler is a summary of the computational model of the brain network models. The level V0 is in fact subsum and in fact the connectivity of the level V1. The spatial support of this is the cortical surface, which is represented by a triangular mesh. The level V2 by a parcellation. The parcellation divides the cortex into different regions of interest. The criteria for this parcellation is really, it's an ongoing discussion in neuroscience, how to divide the cortex. What we have here is the representation of these two, the three levels. The level V0, which is in a node, yeah, the connections between the neural masses, given the dynamics of the node. Then we have region K, yes, that is connected to region J through long-range connections. But the connectivity within one region, yes, is given by a particular kernel, by a particular local connectivity kernel. Yes. The structural data, well, the structural data in fact comes from real subjects. Yes. As I was saying, the connectome that we use, it's considered to be two matrices. One is the weight matrices, which is saying about the strength of the connections between two regions. And the other one is the track length matrix, because, well, there are, the fibers have a certain length, and there is a certain finite conduction speed, and this is important because it will give rise to certain time delays. Now, as I said in the previous slide, where I was explaining about the local connectivity kernels, these local connectivity kernels that are given a certain density of connections or a certain probability of connections in a patch of cortex, yes, depend on the geodesic distance, so the distance along the cortex. And we need to compute this, if you can see, on a convoluted geometry. And the different kernels that can be used, you find this in the literature, and you have the Gaussian kernel, the Laplacian kernel, or the Mexican hat. Every one of them represents something different. In the case of the Mexican, representing lateral inhibition, and excitation and lateral inhibition. There are certain criteria that these kernels should fulfill, but we, in our case, we don't impose any restrictions to build a particular model. People, or if we want to create a particular realization of a brain network model, you can impose the local footprint as you want. There is traditionally, these local connectivity kernels are symmetric and isotropic, and they are said to be homogeneous. Nevertheless, we will see later on that this is not a particular restriction, so we can actually try to have a non-homogeneous local connectivity as well. Why is this important? Well, because there is another type of model that we are describing as well here, and in this general equation that we presented. And these are neural field models. Neural field models, they need a continuous spatial support that is given by the cortex here. And in the literature, we find different spatial supports that go from regular grids to toroidal geometries to the sphere. And the main interest is to understand how the electrical activity propagates on this convoluted geometry. Some examples of these are the work by Girsa and colleagues, is the spherical approximation. So here they evaluated the neural field model on the sphere, and then this was projected onto the convoluted geometry of the cortex. Then we have the approximation we are proposing here, which is a dense network of neural masses, has been described by Spiegler and Girsa, and as well in an upcoming publication. This particular approach has been criticized a bit. We have been criticized. Why? Because we don't include time delays at the local level, which is what Bojack and colleagues did. However, as we will see later, or I can tell you now, in fact, we could actually build a model, including the surface, which has local time delays. It's not a restriction. And then there is another class of neural field models, which include the Laplace Beltrami operator to represent the spatial derivative. And we actually can in our computational model. We are currently working on this, so we will have the implementation of such models, I hope, very soon. Now, all this description, it's important because I hope that you will see now that there is a direct link between the mathematical description and the notation to the computational representation of these models. And this computational gave birth to open source for large-scale brain modeling. So why we need it if we are simulating networks? There are some available. Well, it was due to the scales, due to this multi-scale interaction, and also the treatment of time delays as given by the connectivity matrix. Now, in this particular platform, we can distinguish two types of simulations. The region-based simulations that include the level V0 and V2, that is local interaction of neural masses and long-range connections, as given by the connectome. Or we can have surface-based simulations. We need then the geometry of the cortical surface, and we can include V0 and V1, all the three different levels of structural connectivity, 0, 1, and 2. In the literature here that you have below, the majority of the works are region-based simulations. That is, it's a network as given by the connectome, and some of them included time delays, but many of them they haven't. In the virtual brain, this can be it's up, let's say, up to the researcher to decide the range of the time delays. As I was saying before, I've described many components, many of the building blocks of a brain network model, starting by the population dynamics given by neural masses. I also spoke about the structural connectivity, which is the connectome, the weights matrix given the strength of the connections, the distance matrices, transmission speed, and the coupling term. I also mentioned the cortical surface and stimulation. All these figures here, in fact, is represented inside the library of the virtual brain. This is the core of the virtual brain, which is the simulator. We have another model here, which I didn't speak during the mathematical description, which are the forward models or the observation models. In the virtual brain, we have implemented the forward models for EEG, MEG, and Ball segments, and also the possibility to subsample or resample the neural activity, so we can actually have a data reduction and any type of analysis should be easier. This scientific library was written in Python 2, and here in the case of Marseille, it is possible to use it with the scheduling system OR to launch simulations in parallel. Well, here are some pictures about the sensors for EEG and stereotactic EEG as well. These are the observation models that are implemented in the virtual brain. Now, all the figure that was presented before is now represented here. This is in fact the architecture of the virtual brain. We have the simulator, and we have a main part that interacts with the simulator, so we can so the web interface, so the interfaces with the users can communicate with the simulator. And this language is called data types. The data types are annotated data structures, and inside these data types, we encapsulate different parts of our model, like the surface, like the connectivity, like the simulation patterns. There is another part, which are called the adapters. These adapters were made so the virtual brain can communicate with third-party toolboxes. For instance, we use scikit-learns for certain analysis parts. We use OpenMEG for computing the forward models, NumPy for reading as well the structural data, and something that is not mentioned here is the inclusion of the toolbox, which is the brain connectivity toolbox, widely used for graph-derived metrics. Another part very important in the virtual brain, which is the storage. We have a database that allows researchers to have a full environment to simulate, to store, and to organize their research, and not only actually just the virtual brain. That's why I mentioned that this is a complete neuroinformatics platform. It's not just a collection of scripts. It's already a mature software, and under GPL version 2, like lessons in Linux, Mac, and Windows as well. There is something I forgot to mention in the user interfaces. You can see that there is the web interface and the console interface. We have two main types of users. One, it's experimental neuroscientists or also coming from clinical neuroscience, and we have those which are really brave and like coding and scripting, so we have the console interface. And users can decide which ones they want to face. My main work here has been around the virtual brain simulator and testing, because all this supporting framework for the virtual brain has been developed by the engineer Leah Domide and his group, and her group, and I've interacted with her quite a lot. So don't think that I did all that, because it was not the case. Just a reminder about the two user interfaces. There is the library profile, so users that want just to write the scripts. It's just a Python module. Yes, like any Python module, you can have it in your session and use it and build your models and run your simulations. And there is an alternative profile that also uses a console, let's say, but also has the database enabled and the storage enabled. Let me show you in the interface of this platform, we have every component I've described previously as a module in the simulator and as a building block of the mathematical model is represented in this interface. And users, researchers can select the different parts of the model. They can select which connectivity they want to use. They can set the finite conduction speed. They can use the cortical surface if they want to try new field-like models. Set the parameters for every node. It is not mandatory that you have a network where every node has the same dynamics, but you can start from different dynamics. And what you see on the right is a column where you can see immediately after the simulation your results, time series, or characteristics about your connectivity. This allows you to quickly see the results of the simulations and see what you had is heading in the good direction. There is another part which we haven't described in detail, but you can find it in the virtual brain, is the possibility to build your own simulation patterns. That means that you can study the effect of a certain to the brain. We don't need only to we didn't build this only to study resting state dynamics, but also to be able to study simulations. And you can see here that you can give a spatial profile, a temporal profile, select the part of your cortex that you want to stimulate, and then see the footprint of this simulation. At the end, there are some interactive visualizations as well, so you can see all the activity all around the cortex, different representations. And because the long-term goal of the virtual brain, sorry about that. The long-term goal of the virtual brain is to be an assisting clinical tool. We also provide visualizations that resemble the visualizations that clinicians are used to see, like this. In this case, is the EEG monitor, and we can see the signals from the EEG. There is another very, very important part in the virtual brain, and this is the connectivity editor. In here, you can see the connectivity matrix, which is giving you the skeleton of your model, of your network. And you can decide where you want to study, for instance, the effects of certain lesions. Or one of the applications we have here in the lab, which is not my work, but some people some colleagues are studying is what happens when you don't have a healthy brain, but you have a diseased brain. This diseased brain is showing signs of epilepsy. What the treatment for this is resection sometimes of the diseased part of the brain. Well, in the virtual brain, you can actually simulate the effects of resections. What happens if we isolate a region? All this can be done in this connectivity editor. We also provide visualizations to understand and to study the graph-derived metrics, for instance, with this spring layout. And then this particular representation that I will speak later on. This was an overview of the neuroinformatics platform. The visualization I just show you, the one that had many matrices, corresponds in fact to a delay-resolved structural connectivity. As I say, the structural connectivity is composed of two matrices, the weights matrix and the distance matrix. What you were seeing in every slice, in fact, it was the connections that are at a certain distance. Yes, as I was saying, you have the strength between these two, and then this is the just straight line distance. This, in fact, if you see the cryptographic data, the white matter fibers are not like that. This distance and the finite conduction speed give rise to time delays. And why? Well, there are people claim that a system with a very small delay behaves similar to that for a zero delay. But there is a problem there, and is how do we quantify small enough? When do we know? Not 20 millimeters per millisecond to 100 millimeters per millisecond, depending on whether the fibers are myelinated or not. Besides, the white matter fibers go from 25 millimeters to 250 or more millimeters. So the cortical time delays, or the corticothalamic delays, or the intercortical time delays cannot be really neglected, and we would like to study their effects. In order to do this, yes, there is another motivation behind the study of time delays in large-scale brain networks. Is that the study by Yersa and Dane show that a system with a distribution of time delays, as we have in our case of large-scale brain networks, is more stable than the system with a discrete time delays. How can we represent in a very compact way these two matrices? On one side the strength, on the other side the time delays. And then how can we systematically study time delays with different new models? Well, this one is more easy than the other one because we have a simulation platform and we can run many simulations to understand this. But this one requires first an exploratory analysis of the connectivity matrix. And this exploratory analysis is based on this structure that the connectivity matrix, the box here, can be decomposed in slices, where each slice represents a distance or a certain conduction speed. We're aware that what we're doing here is considering just one average conduction speed, while there could be also a distribution of conduction speed. But as a first approximation, this will work. What we have found with our exploratory analysis is that there seem to be clusters, yes, around certain distances or equivalently time delays, yes, which has led us to think that there are particular subnetworks of characteristic length or characteristic time delays. And we want to study this in terms of dynamics. What I'm showing here is the result of a clustering analysis for 40 different subjects. Yes, the connectivity matrices of 40 different subjects with the dataset that we obtained from the École Polytechnique Fédérale de Lausanne. And we see that these structural subnetworks are consistent across subjects, and there seems to be one that could be linked with the association fibers, one subnetwork that is the corticothalamic connections, and two others that could represent particular white matter pathways. We also try to understand about the interface subnet, whereas for the subnetwork 2, the one that is in the range of 90 to 100 millimeters, there seems to be a bimodal distribution. But this has to be further studied because we could have even more subjects to actually understand where there are two nodes or finite size effects. Some of the results we have is characterized by the mean of the mean length of that subnetwork. Yes, what we have here in this part is the number of regions that the connectivity matrices had: 83, 129, 234, up to over 1,000 nodes. And the means that we found are consistent across subjects and across parcellation sizes. These subnetworks, because we found 4 peaks, can be represented in a compact way as a Gaussian mixture model. And this is the representation we propose to study the distribution of time delays in models. However, we still need to understand what these modes represent in terms of dynamics. And to do that, we will study the effects of a bimodal distribution of time delays or a multimodal distribution of time delays, but this is already the work of some of my colleagues. So, I'm reaching the end. I will go back a bit to what are the benefits, in fact, of having this new informatics platform, of having this general mathematical and computational framework. One benefit, which I'm very interested in, is reproducibility. What you have here on the left are from the 1009 study the stability of a network in the plane of the conduction speed, connectivity strength. Here on the right, although it's stretched in a different way, are the results I got with the virtual brain. And we got exactly the same result, and this is the one I got. So it means that we can put the previous work into one single framework. And why is this important? Because I think that reproducing results and reproducibility is a way for people with simulations. Also, there is something that has been discussed in the past years, is that the bifurcation, what is called the bifurcation points in a network, is not really a point, but is actually a range. A range for the network seems to be at a critical state. And the numerical simulations that can be performed in the virtual brain could help us to understand this particular behavior of large-scale networks. I have some results here for a particular model, a generic oscillator, And what I have here is, in fact, the variance of the time series resulting from those simulations. The transition between a state where all the nodes are in a fixed point and where all are displaying sustained oscillations is not only, it doesn't happen at one particular point, but the network seems to be displaying intermediate states in a range of values for the coupling strength of the network. And here you can see the envelopes of one of the state variables of the model, where we have all the nodes that are in silent states, and here we start having oscillations that increase their amplitude until they reach a point where they don't change anymore. This is the model I used, and the initial dynamics were a fixed point, as I mentioned before. So, to summarize, what we have presented here is a generalized computational model, yes, with a mathematical representation that is transparent, and it links perfectly to the numerical implementation of those models. The implementation in the virtual brain is modular, so if we want to add another model, we can interchange models. If we want to add another structure or another model, we can. And there is something even that makes the simulator something complete, and that makes it a very powerful new informatics, is the supporting framework, yes, that allows users to organize research, to share research, and to reproduce previous work. And there is another point that I forget, is to understand this type of modeling, because the platform so far has been used as well as an educational tool for people who are starting to work with this type of modeling. Challenges and features. This is more related to the new informatics aspect of the virtual brain. As a very project that is already 5 years old, although the initial idea is a bit older than that, we have to keep it working. It's a real challenge, because we have to interact with so many aspects in a new informatics platform. We need to understand the modeling we are doing. We need to understand what the users want. So this is a challenge, but with the contributions that we have, it is possible to do it. There are some other challenges regarding to any software project, just keeping the documentation up to date. So far, we have a vast documentation, and we have produced tutorials for educational purposes. We also have had some contributions from people who are not doing research and from other PhD students in our countries. We have also to improve the APIs so we can integrate our toolboxes better and in a more easy way. And there is another challenge for the future, which is to validate the different neural mass models that we have, or the different models that we have in the virtual brain, and also to develop a certain classification of these models that so far hasn't been done. And regarding the simulator and performance, one of my colleagues, Marmel Goodman, has been working on a GPU implementation to accelerate the simulations in the virtual brain. Also, with respect to the visualization, today we have, in fact, a big responsibility, because with the virtual brain now we have put people in the position that they can generate massive amounts of data just one click away. And we need to take the responsibility to provide means to people so they can visualize the results, and probably they can as well analyze this. With respect to research, which is the purpose of the PhD as well, I found in this PhD more questions than answers sometimes. What I would like to do on my future direction is to keep working on this time delays project describing the space-time structure of the connectivity, and this is something we are doing. Yes, evaluate the significance of the structural subnetworks to know where it is really that there are certain subnetworks that exist. If we can link them to different white matter pathways, as we can observe in the anatomical data. I'm also interested in providing this general model, including the heterogeneous and long-range connections. Evaluate the importance of local time delays. This, as I say, we've been criticized for not including local time delays, but so far no one has compared where or how much they are in, how much they contribute to the dynamics. Although there is here in our lab, we have Andreas Spiegler that is working on something like this. And evaluate hypotheses about the alphabet generation. In the literature, there are several hypotheses about how this algorithm, which is a very important rhythm in the brain, is generated. And one of the theories is that this particular oscillation is produced due to the corticothalamic delay. And in a more technical aspect, I would like to assess the quality of the connectivity matrices. And for this, there is my colleague Timothy Falk, who has developed a pipeline where we can actually change the different methods that are used to derive the connectivity matrices. This would allow us, for instance, to take the data from the Human Connectome project, derive from the same subject, two connectivity matrices, and see how much the dynamics that we obtain change. And these are due to the effects of the tractography methods and so on. And because it was not only my work, to this lab, the virtual brain was already big. It was a mature project, and the virtual, all these people has been working to produce this software. Some that were, I haven't met yet, and some of them that have been my colleagues and my friends here. And the last part is something very important, I guess, for me personally this year, was to mentor a student who contributed to the virtual brain, someone that came up from the University of Perugia, together with the engineer Lia Domide. He successfully implemented the time series. So thanks to him for giving me the opportunity to mentor him. And thanks for the people involved in this project and who put the money so I could work. And thank you to everyone.
Thank you, Paula.
20 minutes.
So we'll start with some questions.
Yes, please.
Regarding your presentation, and we'll start with the Gustavo.
Okay.
Yeah, perhaps let's start with the congratulations. Really impressive amount of work, which I think would be amazing. I mean, I appreciate that most your concept idea. This kind of
Thank you.
So let me start with some general critical questions.
Yes.
And I will start with the very critical one. Because we already involved in the physics. So that's the whole idea of the whole development. The main aim was really to do necessary.
Yes.
So to that extent, the whole platform is relatively easy if I would like to introduce spiking models? Because, as you know, we also are using that.
Yes.
And perhaps people want to also test this type of model. And my intuition as a non-computer scientist is that shouldn't be a problem.
Yes and no. If you want to include another level, then it could be included. If you see that it could be included, it's like just another scale. The type of networks you're working with, they're still networks like this. But what entails more difficulties, or what is a challenge, is that you have to make it compatible with the whole structure. And there is a lot of work about software engineering, so you can put it in a way that is usable, immediately usable. That takes a lot of time. Otherwise, you can add more details. For instance, for a neural mass model, or if you want to describe neurons, at the end of the day, if you see, it's more state variables. It's more equations. Yes, it's exactly. So yeah,
it would be like a neural mass model, but with an incredible amount
of nodes, for instance, and then you start like summing all these different levels. You have to define the new rules of interactions between the different like the it's like mathematical concept is more state variables, a larger and high-dimensional system. I separate then the part of neuroinformatics as software engineering, the engineering that takes some time, so it's
Is it possible to include delays at that level? So it's not the local level, it's like the ultra
low level. Yes. Well, you could use the mechanism that we have for time delays and define, let's say, you have a very high resolution surface, more than 200,000 vertices, then every edge of that vertex could be at the resolution of a cortical cone or even less, and at that level. But it's going to be awfully slow.
Yeah. I would like to have the vision problem.
Yep.
But I would like to introduce a delay in preparation with Is it possible?
No, this is not really possible at the level of the same node. Let's say when you have two populations, those delays are not really described.
It's difficult to extend that because that's even for the necessary.
It would require to restructure a bit the simulator, or just try to use, as I was saying, mechanisms that are already implemented and say what was my level 1. Now, what I was saying, now it represents this microscopic level, but it means that you start hacking it in some things. It would require more work. So as it is now it's not possible.
But the other question that's also for, especially for the clinicians that use this kind of behavior, is somehow to widen, not give them so too much freedom, because that could be very dangerous. If you would select a model,
Yes.
select structure, some others, but then you somehow you have to widen this. The right thing to do is to adjust the parameters, and for each model the parameters will be different. And you have to run always at the end of the day kind of computation without knowing what it is. You have to oblige them to run that, and at least you see that look, I don't know why, but in this vision, for this parameter, the system is fitting my. So that is somehow included. Of course, maybe the tutorials have to be included, but
no, but I think I got your code to No, it's not included, which would be, well, to do some data fitting of these high-dimensional systems is really a lot of work, but to do something like what you do is to adjust the parameters so they better match the empirical data is something we would like to have a way to guide people who are not from the modeling world. They probably don't want to see all the equations, or if you tell them about bifurcation analysis, then they will, what? And it's a concern, all this. You are giving this software, and sometimes people assume that they will click and they will obtain results, but you need to understand, and that's the purpose of the educational courses we are giving. That's also led to the separation of the two interfaces. So the web interface is more friendly, it lets you understand about the modeling without seeing all the equations and implementation. But if you are a hardcore programmer or researcher, you will go and through the command line and try to see the equations, see how all the pieces are interacting.
Even for the user-friendly part, I would use, say, the button saying and the system is intelligent enough, at least for the user, such that when you select it, using one, optimize it, means that you will look in the delay mapping app.
And find the fitting.
If you are using the one, you know, so you optimize on it. At least as a first strategy, of course, there's not but at least you fix you oblige the user not to use any parameter.
Yeah. So to take care of that, first, all the models have parameters that have been extracted from peer-reviewed articles. It's a way to say, okay, they have this particular initial dynamics. Yes. So that's a way to do it. So far, through the web interface, we haven't implemented, let's say, feedback loops. So it means that you run a simulation, compute some metrics, and then, let's say, you fit or you compare with your empirical data, and it adjusts the parameters. But it's something we would like to do. We do have a continuation system.
But you have that at the script level, because you were showing.
Yeah, at the script level, in fact, you can set this feedback loop. Yes, because you can get at each data point that simulator produces, you kind of see that data point. It means that you could perform the analysis, compare it against your empirical data, and then adjust the parameters. So the console interface so far allows you to do this. The web interface doesn't have the mechanism implemented yet.
Right, with that, so for the scientific user, say I would like to work on the script data, and I would like to have much more flexibility, right? And it's not that I am only interested in fitting two parameters. I would like to fit, for example, internal parameters.
Yes.
Each node level or internal parameter of each parameter without clicking. So, in this case, I want to embed this in a kind of optimization algorithm. This is possible.
This is completely possible. Yeah, you can adjust every single parameter of your neural mesh model. We call it vectorize it or spatialize it. For instance, if you have a surface or you have thousands of vertices, you could set a different parameter for every single node in your network. And you can even, we have methods for normalizing the connectivity matrix, which is something that it's done sometimes in some works, which is normalized by the int strength of the connectivity matrix. And we have mechanisms to do this, but as I was saying, there are two parts. One is the implementation, that sometimes is a few lines of code, a few hours of work. Then when you want to give to the community something that is ready to use, then you have to translate all these mechanisms into the interface and something that is just one click away, two clicks away. But it's available.
do you already got some feedback from clinicians?
The virtual brain? Yes, we have one particular case. Is a researcher involved in the virtual brain project, which is Dr. Anna Solotkin. Yes, she is studying structural lesions, trying to fit exactly this, the simulated data with empirical data from stroke patients. It's but as any software, someone is using my computer. As any big software, there is a learning curve. There is so extensive, in fact, and for people who's not coming from, let's say, this particular part of the computational neuroscience community and modeling, there is a vocabulary to learn. Yes, for instance, we try to match, as I was saying, that's why we try to match in the mathematical description to put the same name as we have in the implementation.
And I wasn't really, for me, it was an excellent, I mean, beside the subject.
Yes. Yeah.
So my last question is now you as user, because you have not developed your own this but I know that you test it a lot.
Yeah, part of the work.
So what is your impression? So okay, if someone comes to you, which model? And I'm not picking up. You can use a select. You can use an acceleration.
I was going to say that no one asked me that question before, but that's a complete lie. What I would say, because what we want to do in science is quantify and compare, because through quantification and comparison our models. First, I would like to know which question this researcher or user wants to answer. And my answer, what you can do is to use both models, because you have the possibility. One, oscillations, like a one-dimensional neural mass model, and one that produces oscillations to see the difference. Second, with respect to the parcellation, yes, big debate, as in the work of Zaleski, that, let's say, the graph-derived metrics change as a function of the number of nodes. In the same way, I would suggest that you use a multi-scale parcellation scheme. That is, you evaluate the model with different connectivity matrices, derived from the same subjects, but different parcellations. Yeah, it's more work, but in fact, the connectivity is a parameter in your models. You can say, okay, run this simulation with this same model. Everything is the same, but with these 5 different connectivity matrices.
Well, thank you again for the
Thank you.
Written version.
Thank you. Thank you.
So, so like to congratulate you.
Thank you.
Bringing computational neuroscience to the masses. It's not too bold to say, and I think this is going in the direction of maybe like first cycle. They were MP3 players before, but they were used by geeks mostly. Apple made it possible that the masses and the clinicians in particular started using the technology. And I think this really brings computational neuroscience into a completely different
in space, and some of the questions I have are very similar to the ones that you asked, because now it's changing the ballpark. It's no longer about achieving these simulations. I think that has been the dream for the neuroscience community for the last 15 years. To be able to have something like a virtual brain to model in all its physical detail the dynamics based on the structural connectivity, and really to take in the data that are available both in the health subjects and in the clinical context, and to do exactly the same measurements as you do in the laboratory. And that almost was a purpose for itself. So people, I guess, got bogged down with this goal so much that they focused on it entirely as a goal in its own right, without wondering very much what the next steps would be and what actually follows from that. But as soon as you talk to the potential users, and so, like you and me, we have been talking to the clinicians and we tried to introduce them to the system, you get very practical questions. And I'll try to translate some of these practical questions back into the neuroinformatics. So the first one, very similar to what has been asked before, would be what model to select. So I think the one thing you put in the outlook to actually create a generic model that deals with generic features of neural excitation, let's say, and translate chronic tone into neurodynamics for purposes. I would very much applaud that, because that would really deal with some of the problems that people not familiar with the theoretical space have in selecting an appropriate model. But a very specific question I would have, in the absence of that, I think this would be a great achievement, but also a non-trivial achievement. So I think it would be complicated. The question I have in the absence, current absence of such a model, why did you implement the particular dynamics that are in the system at the moment? So what was their driving point? Why those and how that?
Well, it was many of the models that are implemented were already used in some other works, so we wanted to have something that is already peer-reviewed. The second question is, many of those models were used to model two regions, like very, very simple networks. And our question is, we implement these models. Are these models good for large-scale networks? This is the driving motivation. Is let's use the virtual brain, and where you can plug a model and see if it's good to simulate the dynamics of your brain. If it's not, I think we are free to say, okay, this is not a good model. Let's move on and try to find something better and build on top of that. That was the motivation.
For practical point of view, it is helpful to label them for particular purposes. Many of these models, if you translate them through a slow forward model, will give you exactly the same answer. So for practical user point of view, it's not clear why you should use one than another one. I mean, for a theoretician, it's very interesting because they have different dynamics and compare different parameters. But from the endpoint viewpoint and end user viewpoint, it's not so interesting. Now, there's a difference between simulating slow and fast dynamics, and people need to talk about that, what kind of models we need to. But apart from that, I think it doesn't make so much difference for the end user. And that's why I think it will be very important to have this generic model that encapsulates or embodies many of these generic models.
Yes.
But are there any models that are missing that you think should be there? So there was a feature request for spiking.
For spiking neurons.
What are the integrating fire? What are the kind of?
Oh, you could. What are models? Well, I'm working on in the general representation of a neural field model, which is a bit different to the ones we have here. But still, this is at the mesoscopic and macroscopic scale. You can implement the equations for an integrated fire neuron if you want. With respect to spiking behavior, there are certain models that do produce this spike-like waveforms, and it's the one based on the Maurice-Descartes neuron model, and also you have the Stefanescu-Jersey models, which are have a higher dimension with respect to the others, so they allow you to have more rich dynamics and bursting activity as well. And there is Also the epileptor model that I forgot to mention because it's the work of Victor and Timothy, but it's already implemented as well in the virtual brain, so to study epilepsy.
Feature requests. I would love to have an cycling model, just a simple discrete cycling model, because that to some extent allows the very simplistic study of patterns, and then the virtual brain would allow to understand how these patterns actually translate into medical neurodynamics.
I forgot another model, which is the Hop theory.
So this is something I would love to see. And something related to the question of complexity. So you touched on the aspect of parameter complexity.
Yeah.
And you quantify it simply by enumerating the number of parameters in the model.
The numbers of state variables inside, I'm going to say, of the network itself. Because sometimes we say, you should use a more complex model. What do we actually mean by more complex? In fact, it's because I have more dimensions, I have more degrees of freedom, then I can have different, like the collection of the dynamics is going to be wider. That's why I define the complexity of a model as the number of state variables. It's just a way, but I'm not going to discuss about measures of complexity, for instance.
So I understand that even that measure may be problematic. You think that something like a cellular automaton can be created without any free parameters, and still you get some sort of complexity that's built into the rules of the system.
Because
the entire neuron has certain rules of what you do across the threshold, etc., then you reset. And this adds to the complexity of the model. I mean, this also adds to the computation complexity.
Yeah.
So it's difficult to quantify just by the number of state parameters.
Yeah, it was a way to quantify exactly. More than complexity, I would call it dimensionality. I think that would be more appropriate in this case. And then complexity, you could add, let's say, the constraints that the interaction between nodes gives you as well.
I think it's difficult to say in every case what is a matter dimension of this. Some of these things are quite interesting. Anyways, I'm just saying it's difficult to really put a precise number on it and plot it against some, I don't know, some ultimately it's probably communication.
I understand. It was in fact a bit, that was a difficult part to be able to dissect every model, because sometimes in some works they present you everything in one equation, and I was trying to dissect and saying, okay, we have state variables, then we couple state variables, and we have now a mass, and so trying to give names to the dimensions so we can understand this and then define either complexity or dimensionality. But it's true that sometimes it's not very clear when you have a particular model how the interactions affect the running times, if you are speaking just technical points, how you could fit to that general equation as well. Sometimes.
Another specific point, can you do language specific models? So can you take into account what has been labeled forward and backward?
No, layer specific is, but it could be potentially possible if we could give, for instance, we could have two matrices giving the weights, and you, in this way, you would be giving a functional aspect to the connections, right? Is this the question? Like, because so far we consider that connections are only excitatory, or there is a connection and it has a certain strength, but we don't say anything about the functional character of these connections.
It's often interpreted as some functional character, but as simple as saying this projection terminates and therefore this originates from the other layer. To resolve the local description,
it could be put as a to-do for the future, because in fact, if you have the white matter surface, so the boundary between the white matter and the gray matter, and then you have the surface representing the top layer of the cortex, then you can even add information about cortical thickness. And in that case, we should move toward volumetric models, where you can actually have the distinction of the different layers in the cortex. But we need to implement more data structures and to define the models as well.
Another end user kind of question is, okay, we have measured empirical functional connectivity, let's say. And this is the question I got just 2 days ago from people sitting down with the virtual graph and planning simulations. And then we try to simulate the data. And given the model, how do you define complexity, whether there's a sufficient number of parameters, of course you can also. So their question, what does it mean to match the empirical with the simulated data? I mean, what do I gain from that? I mean, for you as a computation scientist, that's certainly very exciting. That on the screen you have exactly what I measured in the image. But what do we gain from that? And how do we extract some interpretation from it?
Well, it could be seen as validating the, it's interesting for me because for me it would be like validating the models we have implemented, so the simulated data. In the case of clinicians, let's say, okay, you match your empirical data. What if the empirical data is from a sick brain, a diseased brain? Then if you had information about this brain when it was healthy, then you could match the parameters on, or you could see which modifications in your simulated data are necessary to go back to the healthy state. And this is one of the examples we have for epilepsy, that by modifying the structural connectivity, you could go back to stable or healthy dynamics of the healthy brain. This is what we gain, is understanding, let's say, you match your empirical data, okay, that's my working point. It matches this brain. Now I start seeing how this brain can evolve or can change under certain perturbations or under certain changes in the parameters, because some models, they have biophysical units. It means that some parameters could actually represent some very real external perturbation. I've got a question about effects of drugs. What if you can include in your model the effect of drugs, and you could see how your brain reacts to that?
I think generally this is a question of the future, because in the case of small network modeling, we work based on different frameworks that are performing reasonably well. But when you have large-scale models with their many parameters, then of course those frameworks fall down. And so the question is how you actually extract meaningful parameters, given that many of these models give you the same simulated result. So if you simulate all, for instance, Western state code, you can achieve many different models.
Yes.
And they differ entirely in their parameter space.
Yes.
And they all provide near perfect. So the question then is how to extract meaningful parameters. I think this is a conceptual question that goes beyond the virtual world.
Yes, because let's say that you have a collection of models that produce a perfect match for both signals, let's say. But then you start having questions about which parameters or what the parameters of the underlying model represent. And there will be one particular model that you cannot answer any of those questions. Then you need to move forward to a more realistic model where your parameters can actually, or you can actually find the representation in reality of those parameters. As I was saying, in the case of inhibition, let's say local inhibition, or you can add more details about the synapses in this population. So there will be, for me, a set of models that won't help you to answer or to find to draw any conclusions of the brain dynamics, of the local brain dynamics, while other models, which are more realistic, will actually.
I found it very intriguing what you presented about the different connectivity systems.
Yes.
And how you separate them. How do we know this is real? So what's the significance of these separations in the classroom analysis?
The significance, well, it's because I have it was consistent across subjects. With the cluster algorithms, the problem is that you start having more and more parameters to control. You can start with clustering algorithms where you don't have to infer the number of clusters, the appropriate clusters, and then you do. And I tried both. One that is the mean shift algorithm, I didn't put anything, and I got around 10 clusters, but then it was a work of visual inspections. Then I also went and looked at the streamlines of the cartography data, yes. And then just looking at the histograms of the track length, you can clearly see that it's a multimodal distribution. And by the peaks I found in these histograms, I used this number to use the k-means clustering analysis. So of course, I'm setting a certain number of clusters, which varies between 4 and 6. Significance could be assessed, for instance, with the larger data sets, connectivity matrices, with connectivity matrices derived by different methods, and see if we still find these subnetworks. I try to make the link with particular pathways, white matter pathways, but that's more difficult because it could be that you have two different pathways that have the same length. So there, to assess the significance and the importance, or anatomical importance, we should compare the clusters we found with anatomical data. But
you just mentioned that there are non-parametric clustering methods for the significance clustering, and of course you can use permutation-based methods.
And bootstrapping, yeah. This is something for bootstrapping, and there are some indices of Jacquard index. Yes.
I'd love to hear more about that.
Where?
And just verifying that, because you make this big point about the spatial temporal organization. And I love those diagrams. I love these triangles of connectivity diagrams. I think they really just to say that in the real brain, and you also pointed out, there's this great variety of divergence of delays.
Yes.
And even within the visual system, you can have great differences between its visual stream that does object recognition and conscious vision, and the Dawson stream that is localization. Much faster. Localization works much faster in detection of moving objects and figure and so on than identification of color and texture and so on. Yet we always can recognize a blue part that's passing. We have no problem bringing that together. So I would caution that I understand the computation models. A very precise tuning of delays is very important for the brain to be quite so.
Yeah.
And there are also computation models that will completely without delays, as you mentioned, and they achieve for long time to pretty much the same.
Yeah, but well, that was one of the motivations to have this representation as distribution, because in this case, we could, in fact, in the virtual brain, you can set the conduction speed for every single connection. It's not that you are obliged to get one average one, in which case you set the conduction speed and you get, again, a distribution of delays. So if you represent the delays as a distribution, you can perform some analytical study based on the distribution, statistical properties of that distribution.
I would love to find out the visual system, for instance, if that actually matches the
the difference of them.
Thank you very much for all your answers and information.
Thank you for the questions.
And now we want to so I'd like to add my congratulations to those Gustavo. Very impressed with his work on several levels. First with the manuscript, which was very well written and very access so secondly, I guess congratulations both to yourself and to the wider team that are senior connoisseur of open source neuroscience projects. I think this is really software engineering is a very project and very interesting. I like to see this. And turning to your
integration, I think keywords is integration. And I think integration is the next frontier in neuroscience. I think that you're touching on two levels. First, there's the software framework that lets you bring together all these different kinds of data from different modalities and really integrate it all together and check that it's consistent. Also, the data, the models. So I think it's an excellent step forward from that point of view. And then equally at the kind of models, I think you can actually do this general framework, which I think possibly for the first time that you can compare different models and models like so if it's comparing to each other and to the data sets, I think you can decide the software framework and that integration very important. So I had a couple of questions. So one kind of comes from my ignorance of field of user scale modeling. So if I spent my time looking at the topic level, and synapses, networks, and those things. It says somewhere in both you of your mass modeling physics, microscopic systems, but they know independent details of microscopic systems. It's far from taking this is sort of true. Physics, consistent elements have strong short-range interactions, essentially zero long-range interactions. This is completely not true. So I'm going to comment on this justification. It seems it's not clear.
It's at the, let's say, node level. So the interactions of the very small components, which would be neurons, are represented by the mean field or the average activity, and this will represent a node in the network. So these are the interactions that are, say, simplified and reduced. It's not that we are trying to say anything about the long-range interactions. This comes afterwards when we're using this reduction for the node, for representing the node dynamics, and then we start coupling different nodes. I'm not sure if that was exactly a question.
Yeah,
it should represent the node dynamics, so one part of the brain.
So does that mean the assumption is that the interaction of a finer spatial scale than the node, so
Yeah.
are
Yes.
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 So it's true for the cortex and the communication points, for example, and the back of the mouse, so pepper, a huge functional utility, even a very small spatial.
At the very well, that's why, because if you start going down the scale, you end up with something like the Blue Brain project, let's say. So it's a modeling choice to go to the mesoscopic level. In any case, let's say that you have this special support to model cortical cones, or what would be a cortical cones. What you could potentially do is to have different parameters for every cortical cone or these very small populations, and then you are giving some heterogeneity to the larger model. You don't have homogeneous parameters across the entire brain, but actually can simulate different regions or different populations of neurons. Exactly. Well, there is the Janssen-Embed model that has 3 different types of populations, which are actually representing different different layers. Just that we don't have the structural representation of these different layers in the virtual brain. It's just sub-sum in that particular scale of the model.
So my next question was that
Yes. Yes.
so far, and it's huge connections by the regions. So how many comes from how they can sell it to study. I saw some figures showing how good very different results to do it. So
Yes.
how do you what makes them use the functional effects?
Yes, there is now an upcoming publication by Andreas, our Dr. Spiegler. Yes, where he actually is using a connectivity matrix that includes subcortical structures, including a very large and detailed partition of the thalamic structures, and finding the different resting state networks. No, in fact, he's studying dissipation networks. So after stimulus C, which are the networks that are activated. And it's essential in some cases to include the corticothalamic connections to get these networks. If you remove them, then you the number of these networks that you can observe is completely reduced. And that was also my interest to study corticothalamic connections for the last chapter. What happened when we remove them? Yeah, we are working. Yeah. Hopefully next week. I have some work to do.
So second thing part of the difference in conversation between the last models and disposition.
Ah.
So I just really saw the field model as an issue. The big points in last models look very similar like this to the last model. The differences in still have.
Well, to begin with, for the, let's say, a proper neural field model that also include a representation of the spatial derivatives can include an implicit representation of the local time delays. So you have the propagation speed along the cortex. What I was saying, and maybe sometimes is confusing, it's there are different models to understand the electrical activity on the brain. At the implementation level, they will use the same structure and the same mechanism, so we can simulate them. But in, let's say, the assumptions or the theory behind the two models are slightly different. For neural field models, we assume that these local connectivity kernels I was speaking about, that give the connectivity within the patch of cortex, it's homogeneous and isotropic and rotationally and translationally invariant. So it's homogeneous across the cortex. Now, from anatomical studies, we've seen that this is not the case. The local connectivity is different in different regions of the cortex. And the solution we have in the virtual brain is to use this dense network of neural masses. So on one side you gain because you can add some heterogeneity on this local connectivity kernels if you want to study that. On the other hand, what we lose is the local delays. We don't have them anymore. So the level one, yeah, the level one, exactly. So it's one solution. Both types have some advantage that like represent, in this case, local delays. In the other model you don't have, but you gain in being able of representing more realistically the local connectivity. So,
So I one point is having machine cluster. So you can like treatments. So if you say at certain time 1,000 times larger, 1,000 times greater experience. What other things you can do with that? You use that. You use supercomputers.
Supercomputers to.
I'm not saying supercomputers, but from the modeling point of view, it's an interest. Funds for sources, for example.
If I had all the power and I could simulate the brain, well, the question is it's a bit controversial. What do you want to do? Is you want to create a brain as the human brain, or you want to emulate and get the similar signals to what you get from clinical experiments? So our approach is that we say that to get the same signals, the ones that we observe and the ones that are related to behavior, you don't need all the details of the neurons. Yeah. I would have a lot of fun with a supercomputer, but the benefits, well, I'm personally not that interested in the microscopic level, but if that can answer or improve the understanding, let's say, of how synapses work, then no. Yes. Yes, of course. Sorry, now I understand. Yes, there is a big interest because many of these models, in particular the neurofield models or the different approaches for neurofield modeling, they haven't been widely tested because exactly of computational efficiency. So if we had more power, it would be more easy to answer the questions, which is the role of local time delays. So which of these neurofield models is better or better approximates their reality. So this is exactly, yes.
So moving on now to some more aspects. So what I've been involved in the past 2 years is trying to use standardization to get more microscopic fine neurons and to be easier to exchange models. And there are various methods at this level. So what you're doing is the same thing you use
Yeah.
Are there other people who decide to look use standardization?
Not to the best of my knowledge, I'm not aware of that. There is, I know that last year in Stockholm, in the INCF meeting, there was the particular topic of developing ontologies. But the virtual brain, as I was saying, it represents a new informatics platform and this convergence of mesoscopic models. And so far, everything that has been done is been for the microscopic scale, like the open source brain. Although we've seen and we were invited to contribute the first cortical model at the mesoscopic scale and try to describe it in a similar fashion using NeuroML and some other languages to start creating a systematic classification of models. Yes, because this part of the community, of the neuroscience community, interested in mesoscopic modeling is growing. I've seen it during the past 2-3 years that I've been working this. There's more and more people involved. I also think that the classification, like the one that is currently done for microscopic, would be very useful also for non-modeling people. It would be easier to understand and to relate it to, for instance, compare model across the species as well. Yeah. the types of simulations. Uh, well, we haven't benchmarked a lot, but it depends, as I was saying, one of the interests of defining this complexity or dimensionality. Exactly. Exactly. For instance, for simulations, let's say around 30 minutes worth of simulated data for a two-dimensional model. Two-dimensional, I mean the number of state variables in every node. Yes, and using the Hoynes integration method with the time integration step size of 0.2 milliseconds and 68 nodes, you have like 2 hours for, yes, 68 nodes. Then if you have 96, you have a bit more than 3 hours and 30 minutes, and then it should scale a bit linearly with the number of elements. Then if you move to a three-dimensional model, 68 nodes, the same integration method, it's about 4 hours, and that for me was interesting because you say, okay, I'm adding one extra dimension of the local model, and I need the double of time. But then we have to see at the implementation level which are the functions that we are using, because some models have functions which are more computationally expensive. This is roughly the hours. And for an extremely big matrix, like 1,000 nodes, 8 minutes worth of simulation, three-dimensional local model, it was about 4 days. I'm using the ball monitor that consumes a lot of memory. So. Yeah. Yeah. But then that's why I think that there's been a divergence between this more toward realistic modeling of the brain, of the human brain or some other species, and what we know as artificial intelligence. Because the idea is to create smart systems, but we don't want to model every synapse, for instance. We go with simple rules that work and learning systems.
So the one question is about complex stimuli. So all the stimulations are stored in sufficient. See what's simple, you kind of see it close to the key points.
Yeah.
So suppose you wanted to really compare this to say a visual experiment, say a visual experiment, where you have data, say you show someone a motion cloud stimuli, you scan, get a scan, and then showing these motion cloud stimuli. How do you compute those stimulus
I was planning to answer that question during a postdoctoral fellowship, but yeah. So far as it is the virtual brain, let's say through the web interface, we cannot produce such complex stimuli patterns. The first challenge is to represent this rich stimuli in terms of, for instance, frequency, some composition of frequency, and that I think we would need first surface-based simulations. So we have very high-resolution spatial support, and the next step would be to create the map that is representing your simulations to be applied to the model, so to every single vertex, for instance, every single model, and we will have to deal also with the delays, at which time this simulation arrives to the vertex. But I would go with something like this. I would need a mapping between the stimuli and how I can represent this, in terms of what I can represent this stimulation, so it fits the model dynamics. It's very complicated. Yes. By different regions. Well, it's similar to the multi-resolution scheme for the connectivity matrices. So in this case, it's really interesting. Another point is that you could directly have a very high resolution mesh. So and you define the certain connectivity, that is local connectivity of different type of connectivities, which have a different width. I was saying, if I had the computational power you were saying before, that would be an interesting case. If not, yes, it could be possible. In fact, the surfaces, when they are extracted, they have a quite distribution of triangles. So you could have, like, use remeshing methods and have high resolution here or very small triangles. The problem for us is if we want to go to neural field models, where you need to compute this the Laplace Bertrami operator, which is usually the problem when you don't have a very homogeneous triangular mesh to work with. So it's a technical problem that because then your approximation of this Laplace Bertrami operator doesn't converge to the real solution. If you have sometimes big triangles, regular distribution, it's
Spiking systems. It's kind of clear your simulator is probably spiking simulations. How easily applicable simulation engines. So, for example, let's neuron specialized in natural simulations to interface that to the bridge, to use the rest of the framework as before, have the backend simulator be
that's interesting. I couldn't assess how long it would take, because we, as you see, we have a particular structure of every model, and I should see some of these simulators, like network simulators, they also work on they don't have the same representation of the equations and the parameters we have. So to integrate those, how easy, either we should modify our description of the models and go toward, for instance, symbolic representation. In this case, it will be easy to use, for instance, variance. It's a spiking neuron simulator. I've seen these simulators, and there is a difference at the implementation level of how they describe parameters and equations. will require quite some.
I'm not too much work. I'm sorry.
No, I think it's. Yep. Thank you very much. Also, because as now as a spokesperson of the project, I don't think that we want to move like down in the scale, but more to validating other parts like the forward models and this.
So if I may comment on this,
Mm.
we were in the brain project summit, we were discussing the possibility in the neuroinformatics platform some simulators to use like so large scale like mine, sword, etc. The question is when you go up to this level, then you use nest in the context of making highly interesting question for the future. I don't know the answer either, but this will be questions that will be pursued in the brain project because the infrastructure that is in place here for some of the aspects such as possibilities, etc. And maybe very useful. Okay, so I think that's my question.
Thank you.
Okay, so before giving the last words to Victor, I have the opportunity to some words. More words. The question I already asked as a member of the jury, so it's good for the public. First, I want to congratulate you for the work you did for and everybody for the TDB team must be very proud. It must be a milestone now. And but I wish to have to congratulate you for your personal contribution. And the thesis is a long path and it's not straight, you may know. And so you should be proud of you. It's been a good work. I really appreciate that you, the way you, the method you use for your it's very useful for students to use the same structure, and that's great, and shows also your scientific method. So now I have some small questions, and the one aspect is the fact that you can include the delays in the brain, and so connecting neural masses. And my first question would be just very practical. Why is it difficult to include delays in the simulation? Or is it?
Is it?
What makes the inclusion of delays difficult?
Yes, in general, it's memory because it means that you have to storage. You have an array where you are storing, saving all the past states for every node in your network. So if your network increases, then this what we call the history array is increasing size and chewing your memory. That's why it's challenging. Also because the treatment of time delays in the numerical integration is a bit more difficult, although you already have some routines. But for this level of simulations, there was nothing implemented. And what we have now in the simulator is something that allows you to access directly to this history array if you want. So that's why.
So, because that's a very key question in the neuroscience, is the fact that there's some areas where you have convergence, some where you have some kind of fuzziness in the place. So they are very heterogeneous. For instance, in the visual system, in the terminal particle elsewhere, it's very precise, and so very few spikes can converge to the norm and make while lateral interactions are more certain. So, would it be possible to say yes to include that in the code? Yes, but would it be useful to add such a fitness in the delays?
By facilitating like the distribution of delays,
Yes.
or because there are two things. Distribution of delays, we have different times delays, as I was saying. You have a distance for every connection, yeah, between two regions, and you can set the speed. So every connection can have the same speed or not, and you decide. But once that's set, we cannot change that, because it means that your history array represents up to a certain time delay. If you change the speed, then what you have in every element of your array is representing a different time scale. Now we cannot change the conduction speed during a simulation. That has to be a constant. But if fuzziness means like a distribution.
It can be a constant, but for every single connection that can be heterogeneous in the population. That's right.
In the population at the local level, or between regions, because so far we just handle delays between two brain regions. But yeah, so it can be, no, it can be heterogeneous. It means that if you set the conduction speed, it's going to be always the same delay for that particular simulation. It won't change. So far, let's say once you fix the time delay between two regions or two nodes, it's set. So we don't handle that, but if you want to contribute, we have a public repository in GitHub.
The second last question would be, so if you think about now this fuzziness in time and also in the constant space, because you have populations.
Mm-hmm.
So if you consider the trajectory of every parameter of each norm, they make a kind of a cloud of trajectories. So you can imagine that instead of summarizing the view on the activity in the population at a given time by the average rate, you give some kind of a
Density?
like a probability.
Probability? Yes.
Is it something that is used in the community? That you use, instead of using a kind of a deterministic equation, you use a
Yes, I think I didn't speak about noise, but we are not only limited to deterministic systems.
but in a representation, because some representation may be not coherent and some may be coherent. One which will have a large variance and some low variance. Would you describe this variety, the variance
of a certain population? Yes, in fact, with the Stefanescu models, which are you have a, let's say, certain feature of the population, and it's described in fact by a probability density function, and you can control the width of this population. So you are representing heterogeneity at the local level. This
and my last question will be related to one which is also which is to say, starting from the question, when will you be happy with the data that is spit out by the way? And knowing that the fact that professors like Professor Galland, they will look at the fMRI data and try to decode this data to see if you could guess what is inside the brain. So, is there any chance that you have some other plugins? Don't tell me that I have that you will have some kind of a decoder on the brain. Will it be advantageous for
who is willing to do that? Yeah, it's something we have been asked many times to have an implementation for inverse modeling. So far, because of the time and resources it requires, we haven't explored that part. Or, for instance, analysis, like better analysis pipelines. But yes, you don't have to implement it, but someone else will have to.
Somebody could read out.
It's,
Something in the brain.
yeah, well, in fact, I know that some users, they have been using the virtual brain to simulate its data, and then they are using SPM to, let's say, decode. Once we have enough evidence that it's useful, we can integrate.
So thank you for the discussion.
Thank you. Thank you to you.
We move on to a recap. The last word. Start with a question. How you do?
Fine.
2 hours.
Really? Okay.
When Paula came to me, that was about 3 years ago. Yeah.
Much.
Remember, we were sitting in my office and we were talking about starting the PhD thesis in the laboratory. We were talking about some scientific problems, and the problems we basically decided upon that could be most interesting for someone like you was the problem of distributed time domains. Yeah? And we were thinking of paradigms that could be implemented and how to model time-delayed systems. That was exciting at that time, but now look at the trajectory that Paula is taking. She dove into the problem of the virtual brain. So the virtual brain at that time was in its second, third year. And the challenge in the beginning was to basically embrace the neuroinformatics aspect there in order to implement the problem, neuroscientific problem we had in mind. And what did you say earlier? That PhD thesis is never a straight path. So there were many deviations, and I'm actually very thankful to in the last 3 years, because through these deviations you actually addressed very beautifully what is basically represented in all the questions of the community, and I can translate it as integration. It's what we addressed here and what you needed to address in the last 3 years. Are problems from mathematics, are problems from data handling, are problems from coding, and are problems from visualization, etc. Continue this. The challenge there is really to get an integrated view in representation level. For this, you have to be an expert or semi-expert, at least on every single level. You ask about time delays, and there it's numerical algorithms that need to be understood in order to know which one to use. Yeah? And, I wish to congratulate you. You did a wonderful job in embracing this challenge of a field that did not even exist 10 years ago, when neuroinformatics has become, with the words of Klaus Hilger, respectable in neuroscience only in the last 5 to 10 years. So, as director of the thesis, usually my job is to finish up with a few last words, but being who I am, I would still like to ask a few questions. Yes. But I will make it short. It is actually I'm very happy with the questions that were asked here, and because it goes into the direction of integration. Yeah? I think 1 or 2 we can actually dive in a little deeper to see what the difficulty there is. Can you tell me the generic 2D oscillator? What is the default parameter configuration?
Yeah, it's a fixed point, or it's called fixed point dynamics, and it's called an excitable regime.
You just responded to the demand of Klaus Hilger, who asked for an implementation as a new feature in the future of having an excitable system on the network model. It is needed. It is a generic 2D oscillator. This we use on a daily basis, but it needs to make these links come from a totally different direction to actually put these things together. Yes, it is. But very often we talked about the generic Fourier oscillator so often, but we never ever called it exactly system.
No.
We just did it. Never. We just did it for this. But you see, the response of these are the difficulties of integration, because depending on from which perspective you come, it's simply difficult to put your hands on this. You have to step away back. Yeah? Similar, a little bit, but coming from the other direction, the question that Gustavo, that you asked about the microscopic implementation. If we go through this very literally, if we had to go take the virtual brain directly, implement integrate and fire a neuron on every vertex node, let's make it 10 to 8 of them. When we do this, we have to implement also a copy function.
Yes.
How is that being done right now?
Well,
speak a little more technically, just to show people
the coupling function so far we found seen one in the table I presented. They're very heterogeneous because they are mainly model dependent. It depends with what your model is representing. Because so far we have some generic ones, because you could be interested in testing a linear coupling function for any model. But so far we haven't implemented one coupling function for a particular model. So there are not as many coupling functions as models, but there are so far about 8 coupling functions, because it's even a linear function, which is just could be used as a scaling function. Then you have a sigmoid function as well. That's when you need some kind of transformation. These are for the activity-based models, like the Wilson-Italian one. And there is an hyperbolic tangent for the Morris-Lecker-based model, but the hyperbolic tangent is quite similar to a sigmoid, in fact. And we have also the coupling for the Kuramoto oscillator, which is the sign of the phase or the difference, but there's just another one, which is just the difference. And what we would like to have is a more flexible, let's say, coupling module in the simulator where we can build these functions. In the case of, I take the example of the Kuramoto model. You have the sign of the difference. What if we can define the operation difference and then apply another transformation on top? So this is a more technical aspect.
The connectivity kernels.
Yes.
What connectivity kernels do we have? But we'll highlight the significance of
the Gaussian. Yeah. Yes. So the exponential family database and the Laplacian and the Mexican hands.
So we have very explicit connectivity kernels that represent functions, functional dependencies on the discrete, which means if we were to take the system right away and we put a single neuron and then we have the vertex grid, we will have to connect, and it's a translational invariant, this neuron with the Gaussianly decaying away, and then the next one, and the next one. And all this completely isotropic. Is that how real single microscopic neurons are connected? Obviously not. It's probabilistic.
Yes, it's a probability.
So there may be some structure there, but it's probabilistic. And this is the aspect that I wanted to highlight. It would be actually far from trivial to take this and just speed up or increase the dimensionality, because a probabilistic connectivity function to implement it in this and then respond to the great criteria of the vertices, all defined on 2D. You know the difficulty. That would be difficult. So we cannot just tweak it up. Yes.
Top of the Gaussian.
You can, but it has to respect the distribution of the vertices.
Yes.
Yeah, but then it has to be integrated into this large neuroinformatics platform, be consistent with all these
we compute the geodesic distance, and this is another question that what happens if we have a non-homogeneous triangular mesh for computing the geodesic distance? It's possible. Then there is another issue that we should handle. So far we use closed surfaces. What happens if we have open surfaces? we have to start dealing with boundary conditions for these models.
All this is beautifully representative of the challenges that Paula needed to face. Of course, everything can be done, but whenever you change something on a molecular level, it still has to correspond with respect the architectural challenge. And here you would go qualitatively from a functional representation of the connectivity to a probabilistic representation, and that would change quite a lot on the implementation. So these are the hidden challenges you have to deal with all the time. That sometimes not really apparent. Also need the discussion. So this is the aspect I wanted to highlight because I like it very much what you said. Integration.
Integration, yeah, exactly the word.
Thank you for your very integrated.
Thank you.
So, the whole journey has been able to ask a question. So, I would ask to leave the room because we will deliberate.
I have to leave the room as well.
You have to, of course.
You may want to close your computer in order not to broadcast all this action.
Yes. Yeah.
So, the last half an hour, let's say.